Okay, everyone, I think we'll go ahead and get started. I am Rob Burgess, and I'm Chief Business Officer for Sino-Biological. I'd like to officially welcome all of the attendees and our guest speaker today in the next installment of the Sino-Biological Lock and Key webinar series on immunodetection. And today, the title of the lecture and seminar is Rapid Immunodetection in Clinical and Pharmaceutical Applications. Before I have the honor to introduce our guest speaker, I just want to mention one housekeeping issue, and that is we'll be taking questions from the audience at the completion of the seminar. And what I would like to ask is every individual in the audience who has a question, just simply type your question into the chat box. And what I'll do at the end and completion of the seminar is I'll run through those questions and ask those live to our guest speaker. So there will be no questions during the seminar, but afterwards we will be asking them from the chat box. So please put them in there, and we welcome all of your questions. So without further ado, it is my pleasure and honor to introduce to you today our guest speaker. It is Dr. Jeffrey Wang, who is the co-founder and CEO of Novodiax. Jeffrey graduated from Capital Medical College in Beijing initially and obtained his master's in science from Union Medical College, also in Beijing, and obtained his doctorate from Max Planck Institute for Molecular Genetics in Berlin. Dr. Wang also completed his postdoctoral fellowship training at the University of California, San Francisco. Dr. Wang has worked as a research scientist and R&D manager at Biogenics, developing tissue-based diagnostic products. Also as a principal scientist, at Chiron, which is currently part of Novartis, where he was developing antibody therapeutics, and as the director of R&D at Multispan, focusing on GPCR drug screening services. In addition to that, he was chief scientific officer and co-founded Digitab, where the focus was the development of a new diagnostic technology platform. Since 2009, for the last 12 years, Dr. Wang is the co-founder and acting CEO of the company Novodiax. So Dr. Wang, it is an honor to have you today. And I will turn over the screen sharing to you now so that you can begin your seminar.
Yeah, thank you very much, Rob, for the introduction. Let me go straight to my presentation today, since the time is pretty short. So my topic for today is a rapid immunodetection in clinical and pharmaceutical applications. For immunoassay, we know there are many different ways to classify them. So I actually classify them in this way, based on antigen format, assay principle, reporting system, and the method. Obviously, I only listed the most popular assays. If we talk about the assay, it's usually a combination of the multiple categories here. So for instance, if we use a soluble antigen in a direct detection principle, using enzyme as a detection label, it will be a so-called sandwich ELISA. So in another way, if we also start with a soluble antigen using the direct detection principle, using the label-free method, at the end, it can be a vehicle, right? This is another very popular instrument in the in this industry right now. Another very popular immunoassay is a lateral flow. So lateral flow certainly can have a different format, right? Using even either fluorescence detection or nanomaterial. Then, if we come to the insoluble antigen, we know today actually people very often divide the antigen simply in two formats. One called biopsy, another one is a liquid biopsy, right? So for in situ assay or the biopsy, immunohistochemistry is obviously the only immunoassay here. Right. So when we come to the rapid immunodetection or immunoassays, so the most of the popular format everybody know is the lateral flow, right? It's a very popular format. So it's used in even everybody's daily life. The most of the popular one is the pregnancy test, right? But actually today, I want to introduce a new type of lateral flow. So it is a lateral flow assay we developed in-house in Novodiax. Is for the detection of neutralizing antibody against the SARS-CoV-2. So another technology I want to introduce today is for biopsy sample or in situ sample. It is also a rapid assay, a rapid immunoassay. I think this is probably the only rapid immunoassay for the in situ sample detection or immunodetection. We call it actually direct IHC. So let's start with a rapid IHC, rapid immunohistochemistry. So rapid IHC certainly has a very clear market demanding. We know actually during the surgery, so very often the surgeon or pathologist need to know whether the surgery is complete. The cancer has been removed completely, right? That's a so-called cancer-free surgical margin. That's a clear application for rapid immunohistochemistry. So there's also market demanding nowadays for the same day diagnosis and the same day treatment, right? So the critical advantage for the rapid IHC is certainly save time, save money, and eventually save life. Yeah. So when we talk about the rapid immunoassay, very often it also means it's a simple, it's a more convenient, right? It doesn't require a complicated instrument, right? And also for when we talk about the rapid immunohistochemistry, it has to use frozen tissue format. Yeah, it cannot actually run on the formalin fixed tissue because the formalin fixation alone takes usually overnight or over weekend, right? So running a immunoassay on frozen tissue often means we're dealing with a native type of antigen, right? It often result more accurate result. But of course, there's a challenge, right? So first thing is a technical challenge. In order to make it make an assay faster, quicker, we need to actually first increase the assay sensitivity, and then reducing the sensitivity to a level by shortening the assay time. That's the principle we use here. Another challenge is certainly right now, Novodiag is more or less the only one company in this field. So all other large companies in the same field, like a Roche, like a Agilent, like a Leica, their focus are still the regular formalin fixed tissue. That's why a lot of clinical rapid IHC products are still missing. And the last challenge is that because this is a new technology that will generate a new medical procedure. So changing a existing medical procedure is always a big challenge. Yeah. So here is a comparison using Mohs surgery as an example. Mohs surgery is a special type of surgery for skin cancer. So let's take an example here. So on the left-hand side, that's the Mohs surgery procedure using our IHC Direct technology. The procedure actually only takes 10 minutes, a little more than 10 minutes. Yeah. So the very common procedure is that the surgeon remove the melanoma, skin melanoma, and then examine the surgical margin immediately using our technology, our product. So if it turned to be positive, there's a melanoma cell there in the surgical margin, they will do immediately and second surgical removal. Actually, on average, a Mohs procedure takes 2 rounds of surgery and examination. So obviously with our technology, the whole procedure can be done within the same day. The test time in total, 2 rounds of testing time will be less than 30 minutes in our case. But using traditional technology, the procedure also started from a surgical procedure, right? But actually, the clinical sample will be formalin fixed for overnight. The patient will be discharged to home. That's the general, or to hospital, or to to a hotel. That's the common practice. Then once the surgeon receives the test result on next day or even later, they will schedule the second surgery if the result is positive. Yeah. So just to think about this procedure, usually it takes several days. Now you see a clear advantage of using a shorter, rapid immunohistochemistry. So our rapid immunohistochemistry include not only the reagent, the rapid immunohistochemistry itself, also including the instrument. Novodiag will soon deliver the first very compact instrument, the automatic scanner in the market. I think next month, right? So from the left-hand side photo, you can tell how compact the instrument is. If you have the knowledge about a regular immunoscanner, you'll know how small it is. It's more or less, 1/5 of the regular automatic scanner. So here is some example of our available product. We plan to market and sell them panel by panel. For instance, the breast cancer sensitive lymph node panel. For example, in the middle, you can see the Mohs surgery panel, right? and so on. We also have the product line for other cancers like breast cancer and lung cancer. So right now, the breast cancer panel are only available in China. The lung cancer panel is also mostly sold in China, Chinese market. Yeah. So here are some other example available product panel, like a thyroid panel, brain tumor, and the cytology panel. So next thing I want to talk about is the rapid immunohistochemistry in the pharmaceutical application. This is so on this page, we are showing a product we developed in-house. So we actually modified the famous therapeutic antibody, the trastuzumab, right? The Herceptin. So turn the Herceptin itself as a diagnostic tool for itself. So here I'm showing several examples, the photos. So the direct IHC performed on frozen tissue in the middle, on the paraffin tissue on the left-hand side photo, and also the cytology example on the right-hand side. So on the frozen tissue or cytology example, fresh sample, the procedure takes only 10 minutes to perform. We also did actually we did two studies. One was with Brown University, another one with MD Anderson. Actually, in those studies, using Herceptin to do companion diagnosis for Herceptin. Okay, we show actually 100% overall agreement with adjuvant HER2 fish product. Actually, that product is considered as a gold standard in the industry for this application. So other application could be, use a rapid IHC for the cell therapy. We know usually the cell therapy need to re-inject the culture cell back to the patient, right? Currently, the examination method is flow cytometry, but we know there's a market demanding that they want to see the cells by their eyes. Obviously, a rapid immunohistochemistry can perform this job. We also know that our technology can be developed into a rapid cytology company diagnostic panel. For instance, we know right now for the leukemia or lymphoma, right? There are already multiple targeted medicine available, like the drug for CD2030, CD138, right, in the ALK, right, and so on. So think about this. Actually, the company diagnosis can be performed on just a few blood smear, right? So just within 20 minutes, the result can be delivered. The physician can make a decision based on the result. It's a really convenient assay. The same concept actually can be applied to other cancer too. So now let's jump to the lateral flow. This is really a special type of lateral flow for detection of SARS-CoV-2 neutralizing antibody. So talking about the neutralizing antibody, we know that the virus has a lot of many protein antigen, right? The most abundant one is N protein, the nucleoside protein. That's the most abundant protein. Usually, when a company or researcher develop a diagnostic type of lateral flow, they target this antigen, right? But actually, when you want to study the function, the interaction of the virus with a human cell, another protein, the spike protein, has to be the focus, right? On the right hand side, the photo actually shows some of our internal results, testing multiple the antibody against the different antigen. So the yellow curve actually shows the antibody titer against the N protein. Yeah, it's certainly the highest. So in order to actually study the interaction of developing neutralizing antibody, I say, the focus has to be a tiny part of spike protein. So we know actually within spike protein, there's a receptor binding domain. Then within the receptor binding domain, there's a receptor binding motif. Even within this receptor binding motif, there are also two so-called hot spots. So basically the structure or the sequence that can generate the neutralizing antibody is really tiny. So I think talking about the neutralizing antibody, we have to talk about the immunity, right? People usually think about what's the relationship between neutralizing antibody and immunity. Based on our knowledge, so immunity often, composed of two type of immunity, right? One is one is called humoral immunity. Another one is a cell-mediated immunity. So humoral immunity actually for virus is clearly the only neutralizing antibody, right? For bacteria, it also contains complements and some other peptides. Yeah, then just if we think about the function or the mechanism of the immunity, how neutralizing antibody works and how cell-mediated immunity work, they are actually quite different. So neutral antibody is very clear. The function is to block virus from entering into a human cell, right? If you have a protective level of neutral antibody, virus won't be able to enter human cells. But then for cell-mediated immunity, the function is different. It actually, the immune cell detect the cells infected by virus and then destroy the cells, right? So as a result, if you have enough protective neutralizing antibody, you don't develop a disease, even you face the virus, right? Yeah, but then for the cell-mediated immunity, you can assume you don't have you won't develop a severe disease, but something will happen in your body, right? Because your immune system basically need to fight with the virus infected cell and destroy them, right? It's certainly a pathology procedure, right? There's another very important point. although there's a cell-mediated immunity, yeah, it's very difficult to measure, right? But for neutralizing antibody, there are many ways to measure. So now the question is where the neutralizing antibody test has been used and will be needed. So very clear, most of the vaccine developer has used neutralizing antibody test in their clinical trial to determine the, it's actually the more, in many cases, is the only laboratory measurement for the vaccine efficacy. So another important thing we need to understand is that not all the people after vaccination will develop enough neutralizing antibody, right? So when government or public health workers talking about often talking about the herd immunity, that's fine because having the herd immunity developed, we cannot we don't need to worry about a large scale pandemic, right? The pandemic can be controlled. But so at this moment we still don't know how long the antivirus immunity can last for at both the personal level and the population level. Here in this slide, there's the photo or graph from the New York Times. Based on this graph, the prediction is that by the July this year, just next month, so in US, we'll reach the herd immunity, right? Just next month, yeah. But we have to be clear that yes, we can reach the herd immunity. The pandemic can be controlled, but it's not a done job, right? We have to find a way to maintain the herd immunity, right? The herd immunity may one day drop, right? Drop to a level lower than the herd immunity. At that time, we need to have an additional boost, the vaccination, right? But right now, this is still a question mark. We don't know when it will happen, when we need to do the third immunization, right? We still don't know. But in order to do it, we need a convenient neutralizing antibody test to tell. I think it's generally agreed that when we schedule the third boost, it's more or less most likely is a personal behavior, right? Because the antibody lasting time vary between people and the people, right? Yeah, it had to be a personal behavior. So the measurement become more important. So now let's talk about the product. So they are different type of lateral flow product for the SARS-CoV-2 antibody detection. So on the right-hand side, there's a long list. All the assays actually are, those assays are listed in FDA website, right? It's a very, very long list. All of those assays detect either N protein or S protein for the detection purpose. Basically, the purpose of the assay is to tell whether you are infected in the past. Then in the middle, this is something very interesting. Recently, several commercial companies claim they have developed a neutral antibody lateral flow test. But in reality, they are the assay, targeting the RBD domain of a spike protein. It's really not detecting a neutralizing antibody. It's detecting binding antibody to RBD, right? Yeah, we know actually the binding antibody to RBD is not equal to neutralizing antibody. The real neutralizing antibody test system has to contain at least two critical components. One is a ACE2 protein, the human receptor. Another one is the virus, the RBD domain in the virus, right? The neutral antibody actually serve as a blocker, blocking the interaction between these two molecules. You have to have all the components in the system to detect a neutralizing antibody. That's the assay principle for novel DAX neutralizing antibody lateral flow. We call the assay CONAP. Yeah. At this moment, the development is at almost the final stage. So along the development, we already filed a patent, and we also generated several publications, including actually one review article. So at this moment, we know our test can pass stability for 6 months at the room temperature. We know our assay has a satisfactory sensitivity and the specificity based on FDA EUA standard. We still have I think 2 or 3 more assays pending to complete. For instance, number 6, the matrix equivalence study, I think is more or less done, but we are missing one negative example. Yeah, because all of a sudden we have a difficulty to find the negative clinical example. Yeah, here is how the product look like, right? So this is so in one kit we have a 10 tests. So in each test we have the following components. Here is a figure. This is also the figure in our review article in the expert review for molecular diagnostic. So in this graph, it's really very interesting to see how the Pfizer vaccine and the Moderna vaccine perform against wild type virus and the variant beta, the South African variant. Yeah, we can clearly see that so when we use 8 vaccine recipient, they clearly show inhibition against the wild type of virus. But actually, when we turn to the beta, the beta variant, you can clearly see that actually it looked like a Moderna vaccine performed a little better, showing nearly complete inhibition at the condition we used. Somehow my computer is stuck. Give me a moment. I apologize. This is actually the almost the last slides I have. Yeah, actually, I'll stop here because the last slides after this one is a photo. Is a photo showing our team. I'm so sorry because my computer just got started. So the product, this Kona product, we plan to release it just later next month. Thank you everyone.
Great. Thank you, Dr. Wong, for that excellent presentation. I'm sorry you had computer problems at the end, but we still saw a wealth of data there, and it's very impressive what you and others at Novodiax have accomplished thus far. So with that,
Thank you.
to turn over to the questions to start. Hold on one second here. Just trying to get to the very first question that I saw. If I can find it. Just a second. So the first question was from Sabala Dul, if I pronounced that name correctly. And the question is in relation to the competition. There are quite a few rapid IHC diagnostic kits out there from other companies like GeneTex and BioLegend and several of those. And Sabala was just asking if you could compare and contrast the difference between your kit and the others that are out there. What separates and sets your technology apart from some of these other rapid tests?
Okay, actually during the development of this neutralizing antibody detection lateral flow device, we set up a rule for our team that we want to be open. So that's why we publish everything. We really want to make it a piece of scientific work. So the principle we use for this IC is a similar or same as the only FDA EUA approved CPAS, a ELISA kit, ELISA neutralizing antibody detection kit manufactured by GeneScript. We use exactly the same principle. This principle is also the same as the print. That's the plaque reduction neutralization test, the gold standard. The US FDA considered, right? Yeah, we use exactly the same principle, but actually we have been also heard a lot of competitors product. They often claim they are also detecting neutralizing antibody, but when we look into it, we realize okay, it's actually not. They are actually still detecting binding antibody. I think that there's a misleading publications even. I know there are publications talking about the correlation between the binding antibody to receptor binding domain of the virus and the immunity. Yes, the correlation is pretty good, but it's not equal to neutralizing antibody, right? As long as you call it a neutralizing antibody, neutral antibody is only a small portion of a binding antibody, right? So you just consider the structure of the spike protein or even the receptor binding domain. The amino acids or the structure that can contribute or generate, induce neutralizing antibody is a really small portion for the structure, right? So at the time, let's say if you use, you detect RBD binding or spike protein binding antibodies. So I do believe, a tiny percent of the antibody you detected is neutralizing antibody, but the majority not. Right. This is really a confusing message. So at time, actually, there must be the cases that you have a lot of binding antibody against the either spike protein or the RBD domain, but actually you don't have any, or you don't have a protective level of a neutralizing antibody, right? Unless you directly detect neutralizing antibody, is unknown.
Right. You're saying that your system in your kit is the most sensitive or specific on the market for detecting neutralization specific antibodies, right?
That's right. That's right.
Great, great. Question from Wen Hao Yu is very related to the previous question, and it just asked, is the IHC Direct Method of novo diox. It shares the same mechanism with conventional IHC, and it sounds to me like it is the basic principle of other rapid IHC diagnostic kits, right?
I would say, IHC, everybody knows IHC principle, right? What's the mechanism of a IHC? Um, IHC is very complicated. Yeah, I often consider IHC is the single most complicated immunoassay. Simply because it's a in situ assay, right? When you run a in situ assay, you are dealing with basically you are feeding numerous antigens. Yeah, all the cell components, proteins are there, right? So that's why when you do detection, you have to make sure you don't have any you don't detect any non-target molecule, right? So the specificity is really critical. Um, so talking about a technology here in Novodex means that we have a product that consistently deliver the high quality result at a very short time. So right now, by definition, we actually often call our procedure 10 minutes IHC. So that means when we do it manually, if we only run 1 or 2 samples, we can perform the assay within 10 minutes. But actually when we test multiple samples, of course, it takes a longer time. But actually, we also, when we apply the assay on our instrument, so our instrument capacity right now is 10 slides. But when you run IHC, you can run maximum seven IHC, no, 7 different IHC antibody. But the total number of slides is 10. We can actually perform, get everything done within 20 minutes or a little more than 20 minutes, depending on the complication of the marker, difference of the biomarker. Yeah. So, actually developing one rapid IHC is one thing, but then developing many rapid IHC is another thing. It's totally different. I know, actually some researchers, actually even some company had delivered 1 or 2 or a few rapid IHC product in the market. But actually, if you pay attention, they are often dealing with the high abundancy antigen, right? So immunohistochemistry is quite diverse. So here, nowadays, when we talk about the immunohistochemistry, rapid immunohistochemistry, we want to make sure no matter the target is a low abundance antigen or a high abundance antigen, our assay perform the same.
That's great. That really separates you from the competition. The next question is from Rahula Vasek. In LFT, why do we need to conjugate our detector antibody to gold or other agents? For example, in the slide nanobead. Do you understand that question, and could you answer it?
Actually, the audience is asking for rapid IHC, right? It is about a rapid IHC, right? Not for the lateral fluid. Okay.
I think that's what Rahula is referring to, yes.
Yeah, for the rapid IHC, the common practice or the most popular practice is to use an enzyme, simply because the enzyme can trigger further reaction or enlarge the signal, right, by the enzymatic reaction. So the nanoparticle has been used for immunohistochemistry long time ago, but it's right now I think it's mostly restricted to the electro microscopy detection, simply because of the nature of nanobeads. But in terms of very sensitive IHC, in particular detecting low abundance antigen, enzyme has to be the choice, right? Simply because the enzyme has a very good, signal amplification capability.
Enzymes are highly sensitive, and HRP, it's old technology, but it's certainly tried and true, and we've all accepted its sensitivity, right?
Right.
Next question is from Jeremiah Morrissey. Says not all neutralizing antibodies are to the receptor binding domain. There are neutralization epitopes in S1 and S2 regions. Do you plan to test for these as well?
Actually, I forgot to mention, in our system, we actually not like other player in the same field using RBD, the receptor binding domain, as a one party. We are actually using S1 protein, the whole S1 protein. The reason we use S1 protein is that because we know, see, I mentioned in my presentation that there are so-called two hot spots in RBD region that can generate a neutralizing antibody. But actually there's also one hot spot is clearly characterized by a group of researchers that actually even in the N-terminal domain outside the receptor binding domain, there's another hot spot, right? So that can generate a neutralizing antibody. So that's why we intentionally use the S1 protein rather than RBD domain only, the smaller region, to cover the neutralizing antibody against the N-terminal domain. But in terms of S2 protein, yes, it actually also serve the generating neutralizing antibody, but very unfortunately, in any non-cell-based assay, okay, if we don't use the cells, the live cells, there's almost no reason to include the S2, because the S2 serves mostly as a fusion, the fusion function right into the membrane. So if you block this function, of course, you can block the virus entry into the cell. That's one type of a neutralization. But in a system, no matter it's ELISA or lateral flow, because we don't have a live cell, so S2 portion become useless. It only useful when you use a live cell type of neutralization test, like the traditional plaque reduction neutralization test. But that's way too complicated. We all know that whenever you need a live cell in your system, you need a special lab condition, right? Tissue culture condition. Also, more strict safety requirement. It takes several days to perform. It's really not our intention here.
Right.
Thank
Okay.
you.
Great question. I want to switch away from COVID-19 for a few minutes and really back towards cancer, because I clearly can see the implications that your rapid tests have in this field, certainly for surgery. And I want to take a question from a Mr. Jeffrey Reinhardt. And he says, can you predict when your high precision stat IHC Direct Mark 1 test for non-small cell lung adenocarcinoma with same day results on non-fixed surgical specimens would be available to clinicians in the United States? What's your timeline for that one?
Actually right now, as I said, I mentioned in my presentation, we have a 34 FDA registered IVD product, including MART1. So MART1 is available. It's available right now. But the real situation is this: we have been focused on so much on the skin cancer field in the past. So right now we have more or less 200 clinics or hospitals routinely use our MART1 product. But so far, based on my personal knowledge, no one has used our MART1 kits, for other applications. We clearly know there's a need for other cancer, for other application. We will welcome, any collaboration, if you want to apply it, to other application. Thank you.
Great. Thanks for that answer. And I want to ask my own question about the Mohs surgery. I mean, after you started your talk, like almost immediately, I saw the value of this related to Mohs surgery. And you had mentioned that, and you mentioned you focused in on melanoma. But many people get Mohs surgery, including myself, for basal cell carcinoma. Not basal cell, but squamous cell carcinoma.
Mm-hmm.
Have kits that specifically detect that, or have you entered into that market as well? Can you comment on it?
Oh yes, this is when we face the real world, the situation is this. So people actually consider different cancer differently. Okay, let's say in case of a melanoma, it's one of the most aggressive, dangerous cancer. So people really treat it more carefully, right? Compared to melanoma, other type of skin cancer, squamous carcinoma, or the what's another type of cancer?
Basal, basal cell is not as serious.
Right, the basal cell carcinoma. Compared to melanoma, skin melanoma, they are more less aggressive, right? So usually, so that's why just physicians treat them differently. So for instance, for melanoma, the practice before our product available, actually they have to do immunohistochemistry to determine whether the surgical margin is free of cancer. right? They have to be very careful. Even though the procedure is very slow, takes several days, they still do it, okay? Simply because they consider it so aggressive. But then for the basal and the squamous cell carcinoma, because they are less aggressive, so they usually just, during the surgery, um, they still check the surgical margin, but using a chemical staining method, right? You see the difference here. Actually, we do have the product um, for squamous carcinoma and the basal carcinoma, similar type of product, but of course, other biomarker. But right now, right now in the real world, the surgeons only use our product for the most difficult cases. For example, in the cases like squamous carcinoma and the basal carcinoma somehow appear in a form of infiltrating, right? They do not have a single mass, very localized, right? They become, infiltrated. In that case, chemical staining method really cannot tell, cannot distinguish where is the cancer cell, where is a normal cell. In that case, you have to use a immunodetection. That's where our product for squamous carcinoma and the basal carcinoma biomarker used right now, right? They are there. Yeah, the point is that they are there. We do have product, but they are not just as popular as our melanoma detection case.
Yeah, I think you raise a really good point. And I know a number of dermatological surgeons are actually, at least for squamous cell carcinoma, just using morphology under a microscope.
Right.
I feel like certainly you need to be looking at markers and utilizing a technology like Novodiaxis to make sure you get it all, but you don't take too much during the surgical procedure. So your point is very well taken there.
Thank you.
Now at the end of the hour, unfortunately, I wasn't able to get to everybody's questions. I apologize for that, but we will forward them over to Dr. Wang and ask him to reach out individually. I'm going to close this meeting now. And first, I want to thank all of the attendees for attending today. We had a huge turnout. Great questions from y'all. We really appreciate it. We thank you all for staying for the whole seminar. I also explicitly want to thank Dr. Wang. We appreciate your time today and the wonderful seminar that you gave. It's fascinating, wonderful, rapid IHC detection technology. And I personally feel Novavax has a very bright future, and congrats to you and your team on all your existing success. And we wish you all the best of luck in the future. And we look forward to working with you as well.
Thank you. Thank you.
Thank you. Thank you, Dr. Wang.